All right, then we shall begin with this webinar today, hosted by A4Cell, where we'll be talking about how to assess spheroid formation viability using SpheroCHECK technology. First of all, I would like to introduce myself. I'm Antonio Quílez Alvarez, and I'm the head of applications at A4Cell. Here you can get my email address in case you want to further discuss any experimental details, any potential applications you have in your lab to test our SpheroCHECKs. I would be very happy to help you out. And about myself, I completed my PhD in molecular bioscience, working at the Spanish National Center for Cardiovascular Research. Previously, I obtained my bachelor in materials engineering. And regarding my previous experience and my research interest include cell biology, assay development, mechanobiology, fluorescence microscopy, and high content screening. So that's why today I'm in charge of telling you more about 3D cultures and how to apply our SpheroCHECK technology in them. So first of all, I would like to send a message on how we can analyze cells by imagining that we can have an eye inside living single cells. So for this, we have been working with our SpheroCHECK technology that some of you may already know. Otherwise, that's why we're here, so we can get an overview about different topics, and then I'll show you some data about our work in the lab on how to implement this in your current routine workflows. So today, what the topics that we'll be covering would include an introduction to 3D models. We'll talk about 3D culture tips and tricks. So some information that I've gathered that for those of you thinking about shifting from 2D to 3D analysis, some things that you might find useful and also will help you out when setting these new experiments up. So we'll talk about cell culture optimization, different techniques and aspects to take into account for acquiring images and analyzing data, with a special interest in troubleshooting. So what we can do to improve the quality of our cell cultures, the quality of our data, obtain robustness in our assays, etc. Then I'll briefly introduce the SpheroCHECK technology for those of you who don't know what we do at A4Cell. And then I'll review some 3D cell culture procedures with our SpheroCHECK kits and some evaluation that we did in some spheroid models. And to finish, I will talk about future perspectives and some other things we are working on. And I hope you enjoy. So first of all, I would like to begin with this question: what's a 3D cell culture? So I asked myself the same question when I had to prepare this webinar, and what came to my mind was, okay, I can ask the AI. So let's see what they think it's a 3D cell culture. So I asked the AI, how does an organoid look? And this picture represents some kind of mixture between an organoid and an alien, but it seems pretty accurate, having complex tissue functionalities, etc. And then I wanted to ask, okay, but what about a 3D cell culture? And what we can see in these images, it's a mixture between a complex 3D cell culture mixed with bioreactors, microfluidic devices, etc. And then finally, it's how does 3D fluorescence microscopy look like? And this is what we got. So complex tissues with volumetric analysis and multiplexed signals. So once I reviewed this, my next question was, okay, so what are people working on regarding 3D cell culture models? So I searched in PubMed, and what I realized is that in the last 3 years, there are more than 10,000 publications per year in these topics. So and I asked myself, okay, let's see what's the concept of a model, and what we want basically is to create an experimental system that can represent something we want to study. And I really like this quote that says that all models are wrong, but some of them are useful. So we need to find the purpose to fit our model in our purpose. And for this, there have been many different approaches to shift from 2D cell cultures, what people working in cell biology are more used to try to include different cell types in co-cultures, develop spheroids and organoids, and to implement them in 3D solutions that can resemble the in vivo conditions like organ-on-a-chips, bioprinting, etc. So what does that mean? So that means that the level of complexity is increasing. But normally when we increase the level of complexity, at the same time, we are losing throughput. So if we want to move from high throughput screening approaches to complex system, we need to find a balance, right? So let's try to find some kind of compromise where we can have a model that fits our purpose. So the difference between 2D models and 3D models, in this case, tumor spheroids, which is the topic that I'm going to be focusing more on today. We can describe tumor spheroids as cancer cell lines in culture that are quite different looking from what they look like in a tumor. They have different growth rates, morphologies, metabolic activity. They're missing some interactions and some other features that are different to in vivo conditions. So for this, that's why we develop this kind of 3D structures, because cells can be in direct contact in their whole volume with other cells, so promoting cell-to-cell interactions as well as to the extracellular matrix, these proteins that give support to the tissues and that surround the cells. But when working with this kind of models, what we see is that we have regional differences in their activity. As you can see in the image in the upper right corner, you can see how, especially depending on the size of our spheroids, we can have a proliferative ring in the outer part of the spheroid, then quiescent viable zone in the middle, and then a necrotic core. So that makes the microenvironment different at the different sides, because there are gradients that happen in terms of oxygen penetration, metabolites, diffusion, nutrients, etc. And that's why we can have different responses when studying our model. But apart from this kind of simple spheroids, there are some other types of 3D cultures. And probably you have already heard about organoids as well. And the main difference between these two kinds of 3D structures is that organoids, they are normally derived from pluripotent stem cells, neonatal stem cells, or adult progenitors. But the key point is that they self-organize into differentiated functional cell types, which resemble more faithfully in vivo physiological conditions of tissues and organs. And there are different sources to obtain these kind of cells. We can get them from adult stem cells, induce them from pluripotent stem cells, also grow them from tumor cell lines or even embryonic cell types. And then once we have this cell source, it's a matter of finding the right conditions to help them to grow into the appropriate model. So you can see here in these figures how there are many different approaches where we can use extracellular matrix gels or matrices where we can embed the cells. We can put them into suspension in bioreactors to let them grow and produce large quantities of these proliferative cells, or we can put them in cultures with air and liquid interfaces. We can begin from single cells, or we can start to culturing them at different stages, and it's just a matter of finding the right conditions. But a few aspects that are gonna give us more information on why we can work with 3D models to obtain improved results. It's this table. It's the information I'm showing you in this table, where we can see different aspects that differ and explain why we have different responses in the same cell types depending on their cell culture conditions. So first one, it's the cell morphology. In 2D structures, they have a morphology which is abnormal. They tend to spread to be flat. Their thickness is very, very low, and they have some kind of protrusions that normally don't happen in such a large quantity or size in vivo. Whereas in 3D models, the morphology resembles more the physiological conditions. But there are also some other aspects that we should take into account. We can start working with cells in 2D in a few days, whereas 3D models sometimes takes weeks or even months to grow and fully develop and differentiate. So these differences in their physiological state make them more representative of what's happening in vivo in terms of gene expression and the proteome status that they have. Also, 2D models are quite easy to work with because they have a uniform exposure to cell culture medium. We see them in a flat surface, whereas in these 3D models, as I commented, in the different regions that we have, there can be diffusion constraints that cause differential exposure to different treatments, labeling, reagents, etc. But in general, when we are analyzing sensitivity to a drug or a treatment, these 3D models offer improved predictivity. Also, another important aspect, especially when working with high content screening mode or running screening campaigns, large screening campaigns of thousands of compounds, an important aspect is automation. When working with these sensitive structures, the use of automation, robotics can disturb their self-organization structures. So that can disrupt and cause some variability in the data. So we need to be very careful because they are very complex and they are technically challenging. Regarding different stages during drug discovery campaigns and overall during biomedical research, including cell biology aspects, we can see that there are different differently well-identified stages of this process that begin with the target selection to lead generation, lead optimization, then preclinical analysis, and then clinical studies. So for this, we normally include in vitro screen in between lead generation and lead optimization, and then we wait for in vivo validation, and then to move on to clinical studies in the latest stages. So we're going to be focusing in this kind of in vitro screens, where we have already mentioned that we can begin with 2D, easy to run assays, but it's going to make more predictable or much less drug candidates are going to fail if our assays are more robust or more representative when using this kind of 3D models. So the implementation of these 3D models can range from personalized drug development, gene therapy, also for transplantation purposes, also working in drug discovery and toxicology, and in general, disease modeling. So we can study the specific mechanism of a protein, a target, an analyte in our cellular models. So the range of applications is very, very wide. And that's why we want to focus on the advantages and also the challenges to choose the right model that it's going to fit our research. So the advantages of these spheroids is that they have a well-defined geometry. They can produce very nice models for hypoxia, necrosis, invasion, and metastasis in cancer. And also the heterogeneity of the complex tissues can be better mimicked. However, sometimes sample generation is inconsistent. We can have variability because of the lack of automation procedures and very manual protocols that makes very complex and time-consuming to obtain. Also, cryopreservation is an issue and a concern. So these structures, sometimes we need very long times to grow, and we cannot stop them and freeze them and get them back again up and running to increase the throughput and the the suitability of our assays. So to produce this kind of 3D models, there are different strategies that many times include the aggregation of these cells. And for this, you can simply centrifuge the cells and obtain a pellet. So this way, you're forcing the cells to stick together instead of being in suspension. You can also prepare domes or extracellular matrix gels on your Petri dish. You can use hanging drop methodologies, include them in bioreactors, either with shaking or without shaking, microfluidic devices, or the use of particles, magnetic particles to help them to aggregate. Also, regarding the way of using extracellular matrices, you can either embed them in the gel, put them on top of the gels, or prepare this kind of domes that I was mentioning. About the characterization and the classical methods for analysis of these kind of 3D structures. We can use live cell imaging, multimode readers, flow cytometry, and also fluorescence microscopy, which is going to be the main topic here in our readouts and what we use at A4Cell. The assays and readouts that we can obtain from this kind of experiments include the monitorization of cell aggregation. So by simply analyzing the size and shape of our spheroids, also include some kind of functional readouts. We can have either absorbance, fluorescence, phosphorescence, different kind of markers that emit some signal that we can detect. Also analyze cell viability, proliferation of the cells, label them with apoptotic markers to see if they are undergoing cell death, autophagy, and also focusing on mitochondrial analysis, including ATP production, pH, reactive oxygen species, oxygen consumption, and oxygen levels. And also we can stain them to analyze the expression of different proteins with antibodies. So the tips and tricks that I wanted to summarize today after some literature review and our personal experience here at A4Cell is to have in mind always that you are working in 3D. So when working with microscopy images, we tend to be looking at 2D representations, but we need to think that this is just one of the sections or a maximum projection, and we need to think about the whole volume. So that's important when choosing the cellular model, the substrate we're going to use, the sample size, how much we want to let our model, our cell culture grow, and how we are going to be able to analyze it. Also, which kind of platform we are going to use, what objective, and whether we are using confocality or not in our microscope, if available. Because it's very different to have a plastic surface where we have a monolayer of cells that can be 5 microns thick tops. So that's very different compared to an spheroid that can range from 50 to microns to more than a millimeter in the case of the organoids. So for culturing this kind of cells or different models, we can grow them either with monolayer, but also we can use low addition plates, either with U-shaped or rounded at the bottom to facilitate cells to aggregate. They can also be flat, what it's going to improve the imaging possibilities, and it will allow us to work with long distance objectives. And we can also use some kind of spheroid microplates and different solutions that are already available in the market. But once we have chosen our plate, it's important to optimize the seeding density and check how long we want to allow our cells to grow, because it's not the same to start with 40 cells, as you are seeing in these examples with different cell lines, and starting with 10,000 cells. So the homogeneity, it's different, and also it's going to depend on the length, the duration of your assay over time. So regarding the imaging, when moving to 3D applications, it's important to have in mind that new challenges arise. So as you can see here in the image to the right, when working with a microscope, it's important to note if we're working with wide field or we are working with confocal, and we are imaging at different planes and obtaining a clear image of this sample. But also it's important to know how large our sample is, because there's going to be some light diffraction in thick samples, and it's going to be there's going to be some heterogeneity, also increased background. And also phototoxicity to the cells can happen, because we are going to be intensively imaging and applying illumination pulses to our samples that require a lot of energy to be transferred to the cells. So my general advice is to use confocality, because it's going to improve resolution. Also, if you can pre-scan when imaging large amounts of samples to choose pre-scan options, so to have some kind of imaging at low resolution to locate the region of interest where our spheroid is, and then move to a higher magnification objective. So this way we reduce the illumination power and the cycles when possible. As you can see here in these images, wide field images are going to get blurry, especially in over thick samples, and this is going to be especially magnified when using low resolution objectives. So you can see here, it's not the same working with 20x standard 20x objective or a water immersion objective that increases the numerical aperture and thus the resolution of our sample. We can see how after hundreds of microns we get blurry images on the top of our samples. So apart from all these different things that we have reviewed today, what I've realized when doing this review of what's already in the market and the needs that the researchers have is that there's an unmet need for validated assays that specifically interrogate markers in 3D culture models. So that raises a question on how we can help from A4Cell to improve these models and the reproducibility of the assays and the robustness of the assays and the quality of the research in general. Because most of the shelf cell-based assays have been designed for working in monolayers or suspension culture. So traditional monolayers. People are still using the same dyes that started working 30 or even 50 years ago. So we need to develop new tools to adapt to the new realities of cell biology research. So we need to ask ourselves, if the case we are lysing these kind of structures, do reagents effectively do their job? Do they penetrate these dyes or reagents to the center of the spheroids? Is diffusion good enough to allow this? Or do cells block the signal before it reaches the detector because the thickness is too high and we are missing the light excitation in the case of the microscopy sample? So these are some questions that help me introduce our SpheroCHECK technology. Because basically, since the beginning of the company, we have identified that there's a problem of analyzing cells over time, the lack of reagents for live cell analysis, which is especially important when working with 3D structures. So normally this kind of dyes become toxic after 24 hours in contact with our cells. And also they have problems to penetrate inside the cells. Cells also degrade these fluorophores, etc. They have limitations in multiplexing, and they can even get ejected from the cells, and we can lose the signal. So for this, we have developed the SpheroCHECK kits because we have validated that they are non-toxic for the cells. They don't affect cell proliferation capabilities, and they maintain the viability of our cell culture. And we have our fluorescent sensors covalently attached to the surface of the chips, allowing real-time monitorization for weeks. Also, as we have a surface, we can obtain spatial resolution to multiplex different kinds of signal over these solid particles. That they remain in the cell cytosol, and they allow us to analyze the cells, having a proof that it's metabolically stable. So Let me give you a brief overview of what SpheroCHECKs are and how they are described, because the name SpheroCHECK simply means suspended planar array chips. So these are silicon oxide microparticles. They are 3 microns wide and long and 1 micron thick that we can put into suspension and simply culture them and incubate them with the cells, allow them to incubate. And once they are internalized into the cell cytosol, we can start analyzing different features of the cell physiological state. So here, what you can see in these images, it's basically how we produce our kits. Our kits consist of millions of these SpheroCHECKs that we manufacture on top of silicon wafers, where we carve this morphology, specific geometry. We have very tight control of the size and shape of these structures, and these mushroom-like structures allow us to print and functionalize the surface of the silicon with different kinds of sensors, of fluorescent probes. That we can covalently attach, and once we have completed the procedure, we simply peel them off the surface of the silicon and put them in a test tube that it's ready to be diluted in the cell culture medium to give it to the cells. So the best feature of these lab in a cell devices, because that's what we are gonna have, it's the possibility of having a lab inside our living cell, is that as they are non-toxic, they allow us to monitor cells over time and analyze different parameters simultaneously. Here in the graphs below, you can see some data, some analysis, how we have validated that and the exposure of the SpheroCHECKs to the cells does not increase the number of death cells and don't affect apoptotic signaling pathways or mitochondrial activity compared to other fluorophores that you can use and you can find in the market. To work specifically with 3D samples, we have launched the product SpheroCHECK to analyze pH in 3D models. So we can quantify intracellular but also extracellular pH levels by changes in the fluorescence intensity of our fluorescence microscopy images. We have optimized the protocols to work with 3D models to assess the metabolic state of our spheroids and also the viability of those. And the only requirement is that we use an imaging platform that includes fluorescence measurements with enough resolution to locate our 3 microns wide and long chips. So, for example, 20x magnification of objectives. The protocol is quite easy. We can just plate our cells. We can treat with our compounds. We can add our SpheroCHECKs, allow the incubation so they can penetrate inside our cells, and then start acquiring images at different time points. So this is the highest added value of the technology, the possibility of interrogating responses of the cells over time. So we can add different treatments at different time points and simply follow up how these different readouts change over time to gather information on what's happening inside the cells. So now I'm gonna move on to the specific use case that I wanted to share with you today, where we are evaluating spheroids with different SpheroCHECK kits. So in our setup, we included HEC293 cells, this kidney cell line, where we added also the pH kit and also reactive oxygen species kit. For this, we used an spheroid formation multi-well plate from Spphero that then we treated with different compounds, our samples, and we imaged in our microscope with 20x water objective. So the idea was to analyze how these procedures can be adapted to the reality of the necessities of the researchers. So we wanted to know what's the difference or whether it's possible to add the SpheroCHECK at different time points. So we began by adding the SpheroCHECK together with the cells during cell aggregation, and we incubated them for 1 day, and we measured these spheroids. Also, we allowed the spheroids to form and to aggregate, and then we incubated the spheroids with the SpheroCHECK for 1 more day. And then we wanted to have a whole sphere formed of around 200 microns. And then we added the SpheroCHECK and looked at them 3 days later. And what we saw basically is that we could analyze different kinds of parameters and obtain different kinds of data from our experiments. And what we validated is that we could allow the cells to grow and to increase their diameter, their cell number in 3D, and without any effect by the SpheroCHECK, and also that we could locate the SpheroCHECK inside the spheroids in all 3 conditions. So that can be adaptable. You can add your SpheroCHECK during aggregation, at the early stages of the sphere formation, or even when you have a quite large spheroid. Then we did some pH calibration with our SpheroCHECK kit. And what we realized is that there's a dependence on the diameter, because the data obtained for the small spheres that we analyzed with the chips started from the moment of the aggregation, what I called here in the graphs as sample A. We can have a very good discrimination of the different pH calibrators and have a very nice calibration curve, whereas the values get quite closer to non-significant for the model C. That's depending on the time you allow your treatments to go in. Here, basically, for performing this kind of calibration, what we do is to permeabilize our cell sample and include some buffers of known pH. So if we are not seeing a response in this kind of large spheroids, it's probably because we need to allow the incubation to happen for longer times, because as we can see from model A data, our SpheroCHECK are responding well, and we can obtain quantitative data in terms of intracellular pH in 3D structures. Also, we have here the first data of our new kit, the reactive oxygen species kit that we have recently launched. And we observe pretty much the same behavior that we obtained with the pH kit. And as you can see, there are larger differences when we induce ROS production with a treatment of staurosporine, and drug that is known to produce oxidative stress in cells. We can see in sample A how the signal in the ROS kits is increased, whereas in samples B and C, it even decays at at the end. And also, what was surprising is that we obtained larger signals in Model 3. So when having larger spheroids, we are having an increased ROS signal. That can be simply produced because there's no good diffusion of the medium inside the cell culture, and these cells in the inside are stressed in their basal condition. So staurosporin may not be getting diffused inside the spheroid, or even there is no significant effect because the cells are already stressed. So the next thing that we wanted to know is analyzing if the penetration of the chips was good at all the levels, and comparing the different models that we had, adding the chips during aggregation or later stages. And for this, what we wanted to do is some kind of more complex image analysis, and try to segment the different regions. So for this, we did some pre-processing of the bright field signals to locate our organoids, to segment quite nicely our signal, and then identify the different regions in the whole volume. So if you remember from the initial image that I showed you, we can have a core that we are assuming to be necrotic or missing some kind of diffusion of nutrients and oxygen. Then a middle zone, and then the outer shell where we'll have the proliferative cells. So for this, we analyzed the data in 3D, and also we located our SpheroCHECK here. As you can see in the images, we can see how they are well distributed all around our sample, and how they can penetrate even to the core. So in this case, you are seeing a donut because we are imaging for time's sake. We are imaging 80 microns of our spheroids, but we can assume and estimate the diameter that I showed you before to be between 200 and 500 microns, and define these different regions, the outer shell, the middle part, and the core of the spheroid, and calculate out of the total number of SpheroCHECKs inside our sample, whether they are more present in the outer shell, in the middle zone, and in the core. And here, the graphs represent the three different methods that I showed you before, A, B, and C. The chips that were added during aggregation, after the sphere has been formed, or once we have a quite large sample. And what we can see is that there are more chips in the shell in smaller samples. What was surprising for me, and I'm just assuming that it's because I defined the same shell in terms of length, and I should have done it in a relative manner to the whole volume of the cell. But what we can assume is that there is a similar distribution of SpheroCHECKs in all three zones using the different procedures. So we cannot see significant differences between adding the SpheroCHECKs at different time points. Most SpheroCHECKs remain in the middle zone because that's the largest one that we have defined, and we hope we can publish soon some differences in pH and reactive oxygen species gradients that we are still analyzing and further characterizing. So with this, I just wanted to remind you the different products that we have in our portfolio at A4Cell. So kits for measuring pH, different wavelengths, also for measuring intracellular calcium levels. Also our SpheroCHECK product, the very first multiplex kit that measures pH and calcium simultaneously in live cells. And our latest development, the reactive oxygen species kit. Some other things that we are working on and we would like to show you include the next developments that we are about to launch. So the calcium kit in in a longer wavelength because people are demanding it. Also a kit to measure intracellular oxygen, and then the combination of different readouts inside individually multiplexed SpheroCHECKs. Another thing that we are working on is to develop what we call a cell imaging studio, and we are trying to implement cell painting strategies that give us structural information and couple it together to our functional readouts with the SpheroCHECKs to offer a complete solution to the users. And with this, I would like to wrap up for today and just now allow you to ask me any questions that you may have, and I will be happy to answer them. So thank you very much for your attention. If you have any questions, you can type them. Okay, we already have a question in the chat. So what cell types has SpheroCHECK been tested? So far in 2D, we have tested more than 30 different cell lines or different cell types, cells in suspension, cells, adherent cells from human, from mouse, from primary origin, immortalized cell line, tumor cell lines, neurons, endothelial cells. I think we have a document with a list of the different cell types that we can share with you if you want. So I hope that answers your question. Okay, so Rashmi is asking me, my data showed the use with HeLa. I've tested primary cancer cells, protein-derived tumor cells. How's the viability and behavior in those cases? No, not yet. We have limited resources in our lab, and we are just validating and the models that we can have. But we have some users that are already working with this kind of samples, and we hope we can publish soon some results. Okay, let's see. Okay, it was in the QA chat. Do you have any more questions? Okay, one more. Rashmi is asking, have you tested the combination of SpheroCHECK with other fluorescent biomarkers? Yeah, we routinely do so. So we normally include some kind of nuclear dyes and cytoplasmic markers that we add at the very last moment of imaging to help us to segment the cells. And that's no problem. It's just a matter of knowing the fluorescence spectra of our dyes and avoiding overlap. But even if overlap happens, we could even discriminate because of the specific shape of the SpheroCHECKs. Thank you. Are the chips distributed homogeneously along the sphere? Yeah, that was what I showed you in the graph before. we still need to, of course, to perform some more characterization. Can share the screen again. So here in this image, what we see is that we have them. There's they are present in all the areas that we have defined. In the core, there are fewer of them, but it's also because of the size percentage that they represent compared to the total mass. It's way lower. But if we normalize the data compared to the total size or the diameter, we get quite homogeneous distribution. Odile Filiol, did you tested the cell Cytex imager instead of IncuCyte? Thank you. Cell Cytex, no. I'm sad to say that we haven't tested it, but if you have anyone or you yourself want to run an experiment, we will be happy to help you out. Niels is asking, how many chips per cell are typically internalized? That needs to be optimized depending on the cell culture conditions, whether you're working on 2D or 3D. But approximately we get a distribution between 25 to 50%. So we want to do more work in 3D because we think we can increase that number significantly compared to 2D cultures. Rashmi, for an immuno-oncology application, for example, if you have a tumor spheroid formation with a SpheroCHECK and then add immune cells like T cells, how would you envision this interaction? I mean, first thing that comes to my mind is that you can have your tumor spheroid loaded with the SpheroCHECKs, and then add your T cells to the culture. So you're gonna be monitoring the changes that are already happening in the tumor spheroid. Also, you could preload the T cells with an SpheroCHECK kind in different colors, so you can be able to discriminate those two. So that's how I would do it. I mean, I would be very happy to see an application like that. So we are looking for users or people who want to test it out, so and publish. So we'll be glad to discuss. Okay. So if there are not any more questions, I would like to wish you all good afternoon and to say thank you for your attention. So and also for the very nice feedback and all the questions. So thank you very much and enjoy the rest of the day. Bye bye. See you in the next webinar series. Thank you.